Hi everyone, my name's Stelzer-Braid and I'm part of the Virology Research Laboratory in the South East Area Laboratory Services in the Prince of Wales Hospital. And I'll be giving you a lecture on vaccines next week and influenza the week after. And for the vaccines lecture, I just wanted to give you a bit of background on the main types of vaccines and their advantages and disadvantages so that we can hopefully do some more fun things in the lecture.
Okay, so these are just some famous people associated with vaccinations throughout history, and we'll have a quiz in the lecture. And so if you know who these people are, then you will probably win the quiz. So for the lecture next week, I'd like you to hopefully take away three main objectives to understand how vaccines stimulate our immune system and how they protect us from infectious diseases. And number two, describe the different kinds of vaccines available and the main advantages and disadvantages of each type of vaccine. And then apply this knowledge to the development of an experimental vaccine that we'll have a go at in the lecture. So today it's really I'm going to really describe the different types of vaccines and the main advantages and disadvantages. So there are 6 types of vaccination, and the first 3 are the main types of vaccines that we have available to us at the moment. So number 1, live attenuated vaccines, and these are live virus, but they are attenuated so that they don't cause disease. And number 2, inactivated vaccines, where the virus has been grown up and then inactivated by some kind of chemical or heat treatment so that it doesn't cause disease. And then number 3 is the subunit vaccines, where there is a part of the virus in the vaccine but not the whole virus. The other 4 and 5 are experimental vaccines, but they show a lot of promise, so I'll talk about them as well. And number 6 is passive immunization, which is not really vaccination because it doesn't stimulate your immune system, but it does protect you from a disease if you are exposed to a disease. Okay, so first of all, live attenuated vaccines, and these contain a live version of the disease-causing virus that have been weakened in the laboratory, and often they are attenuated through passage in foreign cells like embryonated eggs or tissue culture cells, so that the virus becomes so used to growing in a foreign kind of system that it can't infect humans in the same way as the true disease-causing form. And so they're much less virulent than the original virus. And so they can be used to stimulate our immune system while not causing the same level of symptoms and things than the original virus. And the basis of attenuation in most cases is unknown, since attenuation is achieved empirically, and that means that it's achieved in the laboratory and it's achieved by a method where we passage it through foreign cells, and so we don't exactly know what mutations are important in reducing that virulence. So we don't quite know the basis of it, but we know that it works. So in Australia, the live attenuated vaccines that we use at the moment, some examples are the measles, mumps, rubella, varicella zoster, which is the chickenpox, and also zoster, which is for older people who get shingles. Okay, so the advantages of these live vaccines are, because the attenuated version is pretty much close to the real thing, it activates all parts of our immune system. So our body gets an infection, fights the infection, makes the antibodies. They're easy to create because it's just passaging through cell lines, which is easy, and it's cheap to do that. And it gives a quick immunity in a lot of people, which is, if you have an outbreak of something, then that's really valuable. And it can lead to elimination of the wild type virus from the community, which is obviously something that we strive for when we're talking about vaccinations. So disadvantages, obviously, because it's a live virus, there's a very, very small chance that a mutation could lead to a reversion of virulence. Also, the vaccine strain can spread to contacts of the vaccinee who have not consented to be vaccinated. So, for example, if there's a gastric vaccination, then people who are vaccinated are obviously excreting that in their stool. And if it's a low income or developing country where there's not great sanitation, then that vaccine strain can spread to other members of the community. But of course, that could also be counted as a disadvantage in some communities where vaccination isn't 100% for whatever reason. Live viruses are a problem in immune deficiency disease patients, so HIV, people undergoing cancer treatment, some older people who have lower immune systems, so they can't be vaccinated for these particular diseases for their own safety. Because it's a live virus, it will often require refrigeration, which is a little bit tricky in low-income countries sometimes. The second type of vaccine is inactivated or killed vaccines. This is where the virus has been grown up and then been rendered non-pathogenic or has been killed by chemicals or heat or radiation in the lab. And an example is the flu vax, which a lot of people get. I know I get every year. The inactivated poliomyelitis vaccine and the hepatitis A vaccine. So advantages of these vaccines are that they don't usually require refrigeration because the virus is dead anyway. Killed virus can't mutate back to a disease-causing state because it's dead. So, and there's no way it can cause disease. So essentially, it can be safely used in people who have a lowered immune system for some reason. If they're on cancer, chemotherapy for cancer and things like that, they can still be vaccinated. And the vaccine contains the whole virus, which stimulates our immune system, which is, more effective than having a piece of the virus. But because it's not a live virus, it does stimulate a weaker immune response because the virus doesn't actually multiply in our body. So a large number of virions are needed when you are vaccinated to stimulate immunity. And periodic boosters must be given to maintain immunity. And that's sometimes difficult in communities because people don't remember that they need to be vaccinated, or they move away and the doctors lose track of their people's location and things. So if you need to give people a booster, then it's sometimes a little bit problematic. So also only humoral immunity can be induced, and humoral immunity, of course, is immunity through the activity of B cells and antibodies. Because there's no active infection, there's no cellular immunity. Okay, subunit vaccines. So these are parts of the virus, such as the surface antigen. And a really good example of this is the hepatitis B vaccine, which contains the surface antigen. The chances of adverse effects with these vaccines are much lower, and they're not infectious, so they can be safely given to immunosuppressed people. The disadvantages is that the antigens might not be in their native conformation, so then the antibodies may not recognise the virus when it is actually presented. So that is a bit of a disadvantage. isolated protein doesn't stimulate the immune system as well as the whole virus, which makes sense. You're only producing antibodies against one protein, but a virus is made up of many proteins, so your immune system won't recognize the other parts of the virus. The effectiveness of subunit vaccines can be increased using adjuvants, and adjuvants are allow for slow antigen release because they kind of make the protein bigger, and so it's more difficult for the body to get rid of it out of the cell, so it sticks around for longer. Recombinant vaccines, so fairly experimental, use an attenuated virus to produce viral DNA in the cells of the body. So a good example is the hepatitis B vaccine. So I said it was a subunit vaccine, and it is, but it is also a recombinant subunit vaccine because the hepatitis B surface antigen is produced in yeast cells on a plasmid, and then when the yeast cells produce that surface antigen, it's purified in the lab, and then that surface antigen is injected as a subunit vaccine. So advantages of recombinant vaccines are that the vectors are easy to grow and store. If you're using yeast or some other type of bacterium, they're really cheap to grow and easy, so it reduces production cost. Proteins expressed on a virus, even if it's not the usual pathogen, such as a yeast, they're more likely to have their native conformation because things like yeast are actually really good at putting proteins together in the way that we would recognise them. Cost, a bit of a cost to develop just because you need to get into the lab and look for the right genes to clone, express them correctly on the vector, make sure that the protein is in the right conformation, and purify it in the right way so that it maintains its conformation. And so it does take a bit more time than just, say, passaging a virus through some cells, making an attenuated vaccine. Some of the experimental Ebola vaccines are recombinant vaccines. For example, there's an RVSV ZEBOV, it's called, developed by the Public Health Agency of Canada. And the vaccine uses an attenuated or weakened vesicular stomatitis virus, which is a pathogen found in livestock, and one of the genes of that virus has been replaced by an Ebola gene. And there's actually quite good evidence that is quite a good vaccine construct for Ebola. So it's going through clinical trials at the moment. So DNA vaccines are another really quite new vaccine and are still experimental, although to date they seem to be very effective and their safety record is good. So it's usually a harmless virus into which a gene for a protective antigen has been spliced, so similar to the recombinant vaccines. The protective antigen is then made in the vaccine recipient to elicit immune response. And essentially the DNA is injected into the muscle of the animal or person being vaccinated with a gene gun that kind of blows the DNA into the cells and blasts it in there. And once the muscle cells have taken up that DNA, they actually produce the pathogen DNA, and that stimulates our immune system. And so both humoral and cellular immunity have been induced by DNA vaccines. And a West Nile vaccine is being developed in the US that is based on the DNA vaccine technology. So I think that's something that we'll be using a lot more in the future. So passive immunisation, as I said, it's not technically vaccination because it doesn't really stimulate our immune system, but it involves administration of a specific antibody which has been produced in another human or animal in response to environmental exposure to a pathogen, and then the antibodies produced in that organism are collected and passively injected into the person who has been exposed. And it's used when exposure to a pathogen has already occurred and there's not enough time to induce active protective immunity. For example, if someone is exposed to a particular disease and from which they may die, and if we vaccinated them, they would have to wait 2 weeks for a full immune response to that particular disease. If we give them passive immunisation with someone else's antibodies towards that virus, then those passive antibodies can fight the virus quite quickly, rather than having to wait for that natural immune response. And it confers adequate protection in the short term, but of course antibodies have a short half-life of only a few weeks in serum, and they cannot be replaced except by active immunisation. So we still need to actively immunise that person who has been exposed. So hopefully you now understand some of the different vaccine types and their advantages and disadvantages. So thanks very much for watching. I appreciate it, and I'm looking forward to see you next week at the lecture. Thanks, guys.